Very good morning to all my dear students. So myself Dr. Shibu from the Department of Biotechnology, Safi Institute of Advanced Study. Okay. So in the previous class, we discussed something about nucleus and nucleolus. Okay. So structure and function of nucleus and how the nucleolus is there, what is the role of the nucleolus and what is the role of nucleus and what is DNA, chromosome. These are all the things we discussed in the earlier class. Okay. So today's class is about structure of chromosome. Okay, what is chromosome and how the chromosome structure should be. That's what we are going to discuss today. And let I move to the topic. So chromosome. So what is chromosome? You may be heard about. We are having 46 chromosome, 23 pairs of chromosome, autosomes, holosomes. These are all the things we already know that. Okay. So why it is chromosome? So chrome, it's a color, and soma is the body. So that it is called a chromosome. These are rod-shaped, dark stained bodies seen during the metaphase stage of mitosis when cells are stained with a suitable basic dye and viewed under light microscope. Okay, so it is colored during and it can stained only at the time of metastasis. You can see the chromosome during metaphase stage only. At the time, we cannot able to see it. It was highly coiled and with the help of protein, histone protein. And during the mitosis, at the stage of metaphase, you can clearly see the chromosome under a light microscope. Okay, so the chromosomes were first described by Strasburger 1815 and the term chromosome was first used by Waldeer in 1888. Okay. Next is chromosome number. The number of chromosome is a constant for a particular species. Therefore, there are of great importance in the determination of the phylogeny and taxonomy of the species. Okay, so the chromosome number is important, and it is constant for a particular species. For example, I said human. They are having 23 pairs or 46 chromosome. So Drosophila have different number, and cat is having different, like that. Different organism is having different number of chromosome that will be involved in determining the phylogeny and taxonomy of the species. Okay. Then coming to the morphology of this chromosome morphology changes with the stages of cell division, and mitotic metaphase chromosome are the most suitable for studies of chromosome morphology. Okay, so in mitotic metaphase chromosome, the following structural features can be seen under the light microscope. So coming to the morphology, this chromosome morphology, it is not a constant thing. It may be changed during the stages of cell division. Maybe you may be studied in mitosis. You maybe heard about that. There are different phases: anaphase, metaphase, then telophase, like that. Okay. In each phases, this chromosome morphology will be changed. Okay. So and the better study of chromosome is happened during the metaphase chromosome. That's what it is mentioned. During the mitotic metaphase chromosome are the most suitable for the study of chromosomal morphology. Okay, so in mitotic metaphase chromosome, the following structure, except this chromomere, we can see all the structure under light microscope. Okay, so that's what it is mentioned. Then coming to the structure and region recognized in chromosome, certain structure and region recognized in chromosome. Structurally, and each chromosome is differentiated into three parts. One is pellicle, then matrix, and chromonimate. Okay, like so. What does mean by pellicle? It is the outer envelope around the substance of chromosome. So it is very thin and it is formed of achromatic substance. Certain scientists, such as Darlington and the rest, have denied its presence. They may be saying that there is no pellicle is there, there is no such a substance is present there. They are denied, and other scientists they are accepted that it is having an outer envelope and it is the substance of a chromosome. Okay, that is about pellicle. Once you are seeing the structure, you can able to see what is pellicle. Then matrix. Then it is a round substance of the chromosome in which contain the chromonimate. It is also formed of non-genetic material. So it is a matrix or a ground, like a ground. The substance will support or it's a ground. Okay, of chromosome which contain chromonimate. Then next part, major part is chromanimata. Chromanimata. So it is chromanimata. It is embedded in the matrix of each chromosome or two identical spirally coiled thread. Okay, the chromatinata. So it is having the chromosome. The chromosome is coiled in the chromatic matter. Okay, so the two chromatinata are also tightly coiled together. That they are appeared as a single thread of around 800 angstrom thickness. Okay, this chromatometa are tightly coiled together. That they are appeared as a single thread. You can see only a single thread, but it is a two. Okay, so each chromatometer consists of about 8 microfibrils, each of which is formed of double helix of DNA. Okay, so it's what I mentioned about it. Then coming to the structure, if you are seeing the diagram, okay, this is a typical structure of chromosome at an anaphase stage. Okay, it's an anaphase stage of mitosis. You can see the chromosome should be appear like this. Okay, so here it is having different structure. It is mentioned. For example, this area it is called as secondary constriction, and this area that is centromere area that is called as primary constriction. We can say it is primary constriction, and it is a centrosomes are kinetochore, and here the spindle fiber will attach. That what it is mentioned, spindle fiber, and to say this is secondary constriction, and this is also secondary constriction, and this is satellite region it is having. There are number of non-coding genes will be present, satellite region, and telomere is also present here. Okay, so this is chromatinata. You can see that thread-like structure present here. Okay, so I mentioned that you can see a single thread, but it is of two. Okay, chromatinata. And the matrix, the ground region, it is a matrix, and you can see the matrix which where the chromatinata is embedded. Then the outer layer that is called as a pellicle. Outer layer that is called as a pellicle. Okay, so this is about the structure. You already know the structure of chromosome. Anyway, I am refreshing it. It is the structure of chromosome. Okay, then it is mentioning about the chromatic. Okay, next you want to know about what is chromatic. At mitotic metaphase, each chromosome consists of two symmetrical structure called chromatids. Okay, at the mitotic metaphase, if you are seeing the chromosomes, it will consist of two symmetrical structure that is called as chromatid. Each chromatid contains a single DNA molecule. Each chromatid is having a single DNA molecule. Both chromatids are attached to each other only by the centromere. Oh, I already mentioned that. Okay, so both the chromatids may be attached with the help of centromere, and became separated at the beginning of anaphase when the sister chromatid of a chromosome migrate to the opposite pole. During the phase of anaphase, the two chromosome will be stretched down, and the two chromatids will be stretched, and it will be moving to the opposite poles. Okay. So next is centromere. What is centromere? A part of the chromosome is recognized as permanent, and it is a small structure in the chromanima, and it is marked by a constriction. At this point, the two chromanimata are joined together, where it is a place for the two chromanimata to join. Okay, this is known as centromere or kinetochore or primary constriction. Okay, it is named as centromere, or you can say kinetochore, or you can say that primary constriction, all are same. So its position is constant for given type of chromosome and forms a feature of identification. So based in the presence of this kinetochore, the position is constant for a given type of chromosome and form a feature of identification in each chromosome. So 46 chromosome we are having. Each chromosome, this kinetochore is in a different place. That's why we can identify which is the first chromosome, which is second chromosome, and which is the 46 chromosome. We can able to identify only with the help of the centromere. The position of the centromere is changed from chromosome to chromosome. Okay. so so next is the chromosome are attached to spindle fibers at this region during the cell division. Okay, you already know that during the centromere is a place where the chromosomes are attached to the spindle fiber. The part of the chromosome which lies on either side of the centromere represent arms, which may be equal or unequal depending on the position of the centromere. So, depending upon the position of the centromere, there may be a change in the chromosomes. Okay, there may be a unequal. It may be sometime it may be equal, or sometime it may be unequal. So there are different types should be there. If you are come across with the position of the centromere, that we will discuss in the next class. Okay, so hope today's class is okay for you, and if you have any queries you can contact me. I'll clarify it. Okay, so the remaining things we can see in the next class. Thank you.